Welcome children. Let's crack some genetic problems related to chapter 6, molecular basis of inheritance. I've included under three concepts: Erwin Chargaff's rule, transcription and genetic code. So all possible problems coming under these three concepts. I've prepared slides. So let's tackle some of these problems. So we have the first slide dealing with problems on Erwin Chargaff's rule. So we have to recollect Erwin Chargaff's rule. It states that the ratio between adenine and thymine, guanine and cytosine is constant and equivalent to 1, and it's represented by the equation A by T is equal to G by C is equal to 1. So that should be mandatory when you crack such problems based on Erwin Chargaff's rule. So I'm going to read the first question. If a double-stranded DNA has 30% cytosine, calculate the percentage of adenine. So as I told you, you have to fit in Erwin Chargaff's equation. After that, on the next line, you indicate adenine and thymine on the same line. The following line, guanine and cytosine, and the third line you indicate total number of base pairs as per the question. Now you have to substitute. So with another color I am going to do the substitution. They have given in the question the number of cytosine. Cytosine is given as 30%. Now this percentage indicates the total. The total is 100. So from percentage you understand the total, grand total is 100. So if cytosine is 30, guanine is 30%. Why? Because guanine pairs with cytosine, so the numbers are equal. Similarly, adenine pairs with thymine, so their numbers are equal. So 30 plus 30, 60% is gone out of 100. So remaining is 40%, which should be distributed among adenine and thymine. So 20 goes for adenine and 20 goes for thymine. So now I have settled this problem, and the question has asked calculate the percentage of adenine. So just adenine, the percentage is 20%. These questions can be 1 mark, 2 marks or 3 marks. Depending on it, you should show the steps. For a 3 or 2 marks, please elaborate. 1, you have to put Erwin Chargaff's equation. 2, you have to put these bases distributed A and T together on one line, G and C together on the second line, and total number of bases or total number of nucleotides on the third line. And as per the question, please do the substitution. So you will get marks for every line. But if it's 1 mark, you can directly hit the answer. Now the next, we are going to a little more complicated one, slightly complicated. A DNA segment has total 1500 nucleotides, of which 410 are guanine. How many pyrimidines are present? So the same format, adenine by thymine is equal to guanine by cytosine is equal to 1, Erwin Chargaff's equation. Then I am placing adenine and thymines, guanines and cytosines total. And as per this question is given total number of nucleotides. Yes, they have given the total number of nucleotides as 1500. What else? Guanine is given at 410. If guanine is 410, automatically cytosine is 410. Now we have to find adenine and thymine. So 410 plus 410 gives us a grand total of 820. So from 1500 minus 820, we get 680. So the 680 should be distributed among adenine and thymine, which means 340 adenine and 340 thymine. I hope you have understood the distribution, because the number of adenines and thymines are equal and the number of guanines and cytosine are equal. Now in the question they have asked us how many pyrimidines. So we have to identify what are pyrimidines. Always remember adenine pairs with thymine, guanine pairs with cytosine, and you know the bonds between adenine and thymine, a double bond, between guanine and cytosine, a triple bond. Remember, A and G's constitute the purines and T and C constitute the pyrimidines. So now in the second question, what they have asked is the number of pyrimidines. So you have to go with T plus C. So T is 340 plus 410. 340 plus 410 comes to about 750. If you ask purines, it is A plus G. Clear? So I hope you have understood the second problem, where it is little more complicated because they have asked with respect to pyrimidines. Now the third problem, a segment of DNA has 1000 nucleotides, of which 300 are thymine containing nucleotides. What will be the number of purines and cytosine? So again with the same format, A by T is equal to G by C is equal to 1. This is indicative of Erwin Chargaff's rule. Then A and T's, G and C's total. Now here, as per the question, I am substituting what is given. Thymine is 300. If thymine is 300, definitely adenine is 300, because adenine and thymine are equal in number. So what is the total? 1000. So of 1000, 600 is already distributed. So from 1000 I minus 600, the answer is 400. So 400 is to be distributed among guanine and cytosine. So 200 goes here, 200 to cytosine, and now we have a cumulative check of 1000. So I have got adenine 300, thymine 300, guanine 200, cytosine 200. So we have substituted. Now as per the question, they have asked the number of purines. So what is purines? We have noticed what is purines. Purines are, see I told you, A pairs with T, G pairs with C. So purines are A plus G. A plus G. So substitute. What is A? 300, G 200. So the number of purines are 500. So this also is easy. Clear? So I hope you have understood problems related to Erwin Chargaff's rule. These are the only possible problems. Moving on to the next concept, it's based on transcription. When problems come on transcription, it's usually the mRNA strand that is given to us, or the DNA strand that is given to us. It can be the template strand or the coding strand. So let me start off with reading the question. If the sequence of one strand of DNA is written ATGC, 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 give the sequence of the mRNA, complementary strand, and identify the above strand. So there are three questions based on this. The given sequence is that of one of the strand of DNA. So the tip number 1 I am giving you, first of all you identify which strand of DNA it is. So always remember, the strand which is running in 5 prime 3 prime direction is called the coding strand. The strand running in the 3 prime 5 prime direction is called the template strand. Next tip I am telling you to remember is, always the sequence of the coding strand is equivalent to the mRNA strand, except that T is replaced by U. Instead of T, it is U in the case of mRNA. So they give you a coding strand and they ask you to find the mRNA. You need not track the process of protein synthesis, transcription, translation, recollect and thing. Just remember, coding strand is exactly similar to the mRNA, only thing that T should be replaced by U. So having this in mind, you can work this problem. So now we have identified the strand that is given by the question, is given in the question paper, is it's running in the 5 prime 3 prime direction. So the strand is a coding strand. Now the first part of the question, A, give the mRNA. So they asked me us to give the mRNA, and I already gave you a clue. mRNA, if the coding strand is given, mRNA is just the duplicate copy, except that T is replaced by U. So we'll identify the U's, the T's, and replace it immediately. So we can just transfer it. Then another thing I am telling you, the CBSE is so child friendly that it gives the bases not so much shuffled for you all to find it difficult to transfer. They give it in such a way that one particular slot gets repeated. Here you have identified, if you identify, you can see ATGC is the slot that gets repeated, and you can see ATGC is getting repeated 1, 2, 3, 4, 5. So 5 times is ATGC, but when you see the question paper, you get panic. So you have to transfer, you have to carefully eye to eye contact you have to have in the transfer. No, it is one repeat or one sequence that repeated many times. So they do not do too much of shuffling. Now they gave it as ATGC. Sometimes it may be CCGG or it may be CAGC, and that will be repeated many times. So just analyze the first sequence and you can follow it gets repeated. Usually it's ATGC. So mRNA I am going to transfer, and don't forget the polarity of the mRNA is also 5 prime 3 prime as the coding strand. Now I am going to transfer, it's exactly similar, coding strand is exactly similar to the mRNA. So the same repetition, but T is replaced by U. It is AUGC. I wrote once, and here we have noticed how many times this is repeated, that slot 5 times. So just mechanically you do 5 times. AUGC, 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 AUGC. Verify. 1, 2, 3, 4, 5 times. So I have answered the first question. And don't forget to indicate the polarity. I am giving another color, so you will never forget. The polarity is very, very important. Don't lose marks. Next we shall answer the second part, B. It's complementary strand. So let me erase the scribblings. Complementary strand. ATGC, only this one's complementary. If I find, I can just repeat it 5 times. So complementary to A it is T, complementary to T it is A, complementary to G C, and complementary to C it is G. So remember the sequence TACG, that 5 times. TACG, TACG, TACG. 1, 2, 3, 4, one more, TACG. Complementary strand. And remember, this complementary strand is running in the 3 prime 5 prime direction. So don't forget the polarity. Last question. That is very easy. The first tip I told you when you work these problems. Identify the above strand. So the above strand is the coding strand, because it is running in the 3 prime 5 prime direction. Sorry, it's running in the 5 prime 3 prime direction. That's why it's a coding strand. But if it was the other way around, it is called the template strand. So all possible questions are put in this one slide. So you need not do much reference. You don't need not do any reference. This one, you try to work out one problem. This will help you for solving any problems coming under transcription for the CBSE 12th. Now moving on to the last concept, problem based on genetic code. This you have to remember the 6 salient features of the genetic code, and then only we can work out this problem. Here usually in the question they give the mRNA strand. See what is given in the question is the mRNA strand and the protein that is translated. I will read the question. Identify the salient features of the genetic code giving reasons by studying the following nucleotide sequence of the mRNA strand and the polypeptide translated from it. So you have to infer from this, you have to infer the maximum all the features of the genetic code and give reasons. So my first tip for you all is, when you see this strand, see this is the mRNA strand, you can recognize because it runs in the 5 prime 3 prime direction, and also they have already mentioned in the question paper that it's an mRNA strand. So first of all you identify the starting codon. So coincidentally the starting codon You are familiar with the starting codon. What is it? It is the initiator codon. Next thing what you have to do, identify the last codon. Here also coincidentally or purposefully I put it is the stop codon. So, we got our two features. So, the first feature AUG. What is the significance of AUG? It is an initiator codon and codes for the amino acid methionine. So, you got one of the features of the genetic code. Second, you got it from the end, UAG. Do you know what is UAG? It is a stop codon. There are other stop codons, you should also remember them. They are UAG, UGA, UAA. It is the stop codon and do not code for amino acids. So, you got the feature. Third, you can recognize a lot of U's and you see what is these U's. Triple U's coding with phenylalanine, and that should strike you. U is a universal codon. What's the meaning of universal codon? Right from man, who's very complicated, to bacteria with a simple body design, has UUU coding for phenylalanine. So, you got that feature also. Then another feature you can see is, you look at this, the same amino acid phenylalanine is coded by different codons. Look at the codons UUU, UUC, UUG, UUA. So, this explains the feature called degenerate, which states that some amino acid, here the amino acid phenylalanine is coded by more than one amino acid. So, here you can see this phenylalanine is coded by more than one codon, that is UUU, UUC, UUG, UUA. So, here you can see the very same amino acid is coded by different codons. Once degenerate is there, automatically you can blindly go for specific. You need not refer the genetic code to make it specific. For to understand whether it's specific, you have to make a reference to our genetic code, which is on page 112, but it's already, we know the information that our genetic code is specific. The meaning of specific is codons, strictly codes for one amino acid. If you cross check the genetic code, you can't see UUU coding for any other amino acid like leucine, lysine, glycine, arginine, etc. It only codes for phenylalanine. AUG codes only for methionine. Certain, they are only specific, they do not code for anything else. So, here you can give UUU codes for phenylalanine to indicate the feature specific, that is codon codes only for one amino acid. So, we have incorporated nearly everything, but one more feature which is left. Let us check whether we can incorporate that or not. So, when you cross check, we got the initiator codon incorporated, we got the stop codon incorporated, we got the universal codon incorporated, we got degenerate, and when we incorporate degenerate, automatically please incorporate specific. Degenerate and specific. So, what is left? We have left one feature, that feature which states that mRNA is read in a continuous manner. Without any punctuations, without any pauses. So, we will cross check whether in our question they gave it as per the genetic code. Yes, I have erased the scribblings and you cross check whether it is given in a continuous manner. Yes, it is given without any punctuations or pauses. continuous, A, U, G, U, 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 C, U, U, G, U, U, A, U, A, G. When without pausing I read it. So, no gaps, no punctuation. So, I can apply this. mRNA is read in a continuous manner. So, I have incorporated all 6 features of the genetic code, and such a question can come for 5 marks, which means that try to incorporate all the features of the genetic code. Somewhere or the other it can be applied. Now, related to this particular concept, I have another question. The question is, they will give you this sequence and they will ask, this will not be given, so I am striking it. With this sequence, they will ask you how many amino acids are present in the protein that is going to be translated. So, you count the codons, 1 demarcated, 2, 3, 4, 5, 6, 6 codons. Does 6 codons correlate to 6 amino acids? No. In this case, we see the last codon, UAG, do not code for any amino acid. So, here though there are 6 codons, how many amino acids are found in the proteins? Just 5 amino acids, that you should remember. Then they need not give you a stop codon at the end. I am rewriting, it can be any other codon. So, in that case you can go correlate with the number of codons to the number of amino acids, 1, 2, 3, 4, 5, 6. So, you have 6 codons and you can easily get 6 amino acids. So, where did you focus at the end, whether the ending is having the stop codon or not. So, such questions are asked, not so easily asked, a little more with a bigger number, like say 45 codons, how many amino acids, or 45 bases. So, you divide it by 3. Okay, they will give it in triples, in multiples of 3. If suppose they say 27 bases are there, how many codons, how many amino acids, clear. So, always if they say if it is a full fledged protein, it starts with a start codon, ends with a terminator codon or a stop codon, which does not have an amino acid. So, if they say a protein is formed, definitely if there are 27 amino acids, 27 bases, we just divide it by 3. 27 divided by 3, what is the answer? You get 9. So, remember how many codons are there? There are 9 codons. And how many amino acids? Remember 8 amino acids. Why? It is a full fledged protein, which means there is a start is the methionine amino acid, and the end there will be the stop codon, which do not codes for any amino acid. If they are not giving the sequence, if they giving the sequence, you have to cross check the end. But if they are not giving the sequence and they are just telling a figure, like say 27 bases are there and this gets translated into a protein, how many amino acids are present? So, you do 27 divided by 3, you get 9. So, you can say 9 codons, but 8 amino acids. I hope you have understood why 8. The last codon do not code for any amino acid. So, it is actually easy, it is what we learn in the genetic code should be applied. So, it can either the number of codons or number of acid questions, amino acid questions can be asked by giving the sequence or by giving the base number. When they give the base number, be very, very careful, very, very prudent that the last codon is not coding for any amino acid. But if they give the base, check the last codon. It need not be stop codon. It can be a codon that codes with amino acid. So, be very alert. I hope you have understood the problems coming under these 3 concepts, and you can easily tackle such problems in the exams by saving time. Okay, thank you.